So, Eric, thank you very much for just doing it by the seat of your pants. That was just fine. Eric said, "I don't have anything. Someone asked me to do this at the very last minute." So we reminisced for a moment about a famous Clint Eastwood movie called The Outlaw Josie Wales. And if you've seen this movie, at one point Josie says goodbye to one of his fallen comrades, and as he rides away from the gravesite, his parting words are, "I rode with them, and I got no complaints." So, concise is good. I have the fun topic today of 5 new things in clinically relevant neuroscience. This is a talk that I gave a year ago. Obviously, I'm not going to talk about the same things. It was a lot of fun. And I was looking forward very much to doing this talk. But I have to just be honest with you. I've got a disclaimer, and a pretty serious one. My dog ate the homework. That's essentially what happened to me. My computer blew up. And I mean, the whole motherboard, everything. And that happened last Saturday with a little flooping around. Adrian finally took it to the computer burial grounds on Tuesday, and it left me trying to get things off of the backup system, find a computer that I could work with. And so things may not be quite as mellifluous and as well organized as I would have liked. So I had fun last year pointing out a couple of things at the very outset. One is there's almost nothing in neurobiology that isn't potentially relevant to neurology. And if we all were able to live long enough, I'm pretty doggone sure that virtually anything I could bring up would find relevance given enough time. I had fun last year by pointing out to all of you that our government is a little bit interested in some of the things that we might be working to discover. And they've actually met and talked about this, and they're planning to keep their eyes on us, especially with regard to this, they say here in this special high security meeting that they had, that we should, they want to monitor the intelligence committee should be monitoring our discoveries in neurobiology, because we might be coming up with thought control, and who knows what. I know that's something Tom Bird's always thinking about. He wants the gene for thought control. So here were the 5 new things, clinically relevant neurobiological things for you, this group of academic neurologists that I decided on. And I've given them titles here. Our Sexy Brains. I thought that would at least keep a few of you in the audience briefly. I want to talk about how oligodendrocytes myelinate axons. And obviously, myelinating axons is a very important thing clinically. Oligodendrocytes in learning, how do axons eat, and then I was going to throw a little pop culture at you. But with this problem I had with my computer, something had to go. So let's start with our sexy brains. And let me say that there's a new face on the scene who I did not meet until about a year ago, Margaret M. McCarthy, who's doing extraordinary work on the sexual orientation, or the sexual differentiation, I should say, of the brain. And it's coming down to some very hard and interesting science, and I want to share a little bit of that with you today. It's been a much debated topic whether or not there are big gender differences in our brain, and it hasn't been until the 60s and early 70s that people began to realize that indeed the brains of men and women are structurally and very likely, at that point it was mainly speculation, were functionally different. One of the places where there is unquestionably a great deal of sexual dimorphism is in an area called the preoptic area, and especially an area within the preoptic nucleus called the sexually dimorphic nucleus of the preoptic area, or SDNPOA. And what you find there, and these changes begin to happen and are in evidence in a very young juvenile mouse, that in female animals, this crucial area of the preoptic area, the sexually dimorphic nucleus, is much smaller than the similar nucleus is in men. And the other thing that's noticeable and really caught my attention and now has everyone looking on is that there's some interesting differences in microglial cells. So that's where we're going. I'm going to talk a little bit about what we know, how this comes about, and what its implications might be. So first of all, I've mentioned that the nucleus in this base of the brain, in this very highly important area of the brain, is 3 to 5 times larger in males, and it also contains more of a special kind of GABAergic neuron that's identified by a calcium binding protein called calbendin 28K. And the interesting thing about calbendin 28K is that it has been found in some instances to be neuroprotective. I don't know much about that. Maybe my son, Chris Ransom, might know something about that, but that's as much as you're going to get from me. This difference that I'm pointing out here is very clearly related to testosterone, and it has to, the testosterone influence has to occur at a very early time. Part of it is prenatal, and then maybe for a very short time postnatal. In addition to the things I've already mentioned, I'd like to also point out that in males, there's a distinct difference in the appearance of microglial cells. They appear to be a little bit more in the form of what's called a activated microglia, whereas in the female mice, it's in this ramified form, which had been called resting and is now called the surveillance form. So to kind of go over this once again, microglial cells enter the brain at a very early stage. They populate widely, and it turns out that we now clearly know that there are simply more microglial cells in the male brain than in the female brain. Originally, this was an observation confined to the preoptic area, and now it's believed to be more general. There is no consensus about how this comes about. Is it because more microglia are created originally, more migrate in, fewer of them die as a result of a developmental apoptosis? We simply don't know. There's been an enormous amount of effort to understand this dimorphism, this gender dimorphism, and a lot of progress has been made, much of it by Margaret McCarthy. Here's what we know in very crude summary. Testosterone from male gonads acts at a critical time, and it needs to be converted to a much more powerful effector estradiol. If you block that step, then this effect on gender dimorphism is partially blocked, in some instances wholly blocked. There was years spent searching for neurotransmitters that were going to be specially manufactured and perhaps in a unique way distributed in the brain to explain the subsequent changes, they were never found. It's now believed that the estradiol works on COX, these special enzyme systems that produce prostaglandins, and that especially it produces prostaglandin E2. Prostaglandin E2 had effects that seemed very powerful. A single dose of prostaglandin E2 was equivalent to a whole week or two exposure to testosterone. And people couldn't figure that out until they discovered that a single dose of PGE2 produces a feed-forward enhancement of intrinsic prostaglandin E2 synthesis, and that synthesis is carried out by microglial cells. So the microglial cells do this, and then there's a step that's believed to involve glutamate, and very recently, very good evidence that there's also a necessary involvement of astrocytes. And we get out of this several effects, which include many more spines, 2 to 3 times more dendritic spines in places like the hippocampus, at least for a period of development, a much higher concentration of microglial cells in a number of areas of the brain, not just restricted to the preoptic area. And if you interrupt this, you interrupt the development of what ultimately come to be recognizable as male traits versus female traits. In looking at this difference, which I pointed to briefly, that the males during early gender dimorphism have these microglia that look as if they've seen an inflammatory stimulus in comparison to females, which have, even though it looks very elaborate and you might have been inclined to call this the activated microglia, indeed it's called the resting microglia. And in completely healthy normal brains, we see microglia that look more like this. During development, males have much more of a microglia in this amoeboid form, the sort of quasi-activated form, and females, this particular form. Now, those are, in a very condensed version, some of the new facts about the gender typing of the brain. And you come away from reading this literature, even superficially, as I've done over the last several days, incredibly impressed by the robustness of the science. And if there was anybody in the room who was on the border of whether or not there are big differences or aren't, I would urge you to see for yourself, but I'm convinced that there are huge differences, and it now boils down to things that are very solid science. So what more might this mean in terms of clinical relevance? A paper is in the what we call early view, which means it's been accepted and you can look at it online in the journal Glia. It just, I think, reached that stage a couple of weeks ago, which took a look at a Spanish group, sex differences in glia reactivity after cortical brain injury. And here's what they found. So they delivered in females and males a cortical stab wound. These are adult animals, probably about, if I recollect correctly, something like 6 weeks of age, so they're sexually mature animals. And then they looked about a week later at the histology. They binned the brain by distance from this cortical lesion. So we have in bin 1, the very adjacent area, bin 2 further away, further away, further away. And they looked using a stain, IBA1, which is a pretty good marker for microglial cells. The experts in the audience will point out that you can't probably stain them all with any of these markers. You have to be a little cautious about that, but this is a pretty good marker. When they looked quantitatively at this, they saw some things that are really quite interesting. And among them was the fact that close to the lesion, there were many more of these microglial cells in males than there were in females. So I think that's now on your screen. And interestingly, the males, which had more microglia, had microglia that were predominantly in the non-reactive morphology. They did a number of other tests to try to demonstrate that, in fact, this is the anti-inflammatory form of the microglia that they were looking at and seeing more of in males compared to females. So that's quite interesting, and was one of the very first bits of evidence along that line that I had seen. They took a look at neurons, and they did the same thing about a week after the injury. My recollection is that this is a new stain, and I think, and I actually am a little embarrassed to say that I can't remember which is the female and the male in this case, but let's just look at the quantitative stuff down below. And what you can see down below is that there is very clearly in the males a preservation of neurons. So same injury, same timing, lots of robust data collection, and for this one particular type of injury, very clearly, even though you have, well, not even though, there are more microglia of the anti-inflammatory type that appear in the male brain, and the total extent of neuronal loss is greater in the female brain. So to sum this up, I think this is a fascinating, potentially unbelievably important nuance to the way we've done scientific experiments involving the brain. Frankly, a lot of us were quite put off by the NIH's demand that in the future, all preclinical research be done, demanded that it be done on equal populations of males and female animal subjects. There were passionate editorials which were scathing about what a waste of funding this was going to be, et cetera, et cetera. I must say that this kind of data makes me think that they're probably on the right track. Very likely not for everything, but certainly for some things. So we talked about these gender-brain differences, the importance of glia in the generation of these differences, and then the suspicion that there's going to be differences in how male and female brains react to certain damaging stimuli. Now, if there's anybody in the room who, yes, Eric. Eric, I think, one hopes that all of the politics and the sensitivities will be wrung out of these important observations so that we can pursue them in a thoughtful fashion and bring data to the table. I don't dispute that what you suggest could well turn out to be the case. It's a long way to get from where we are today to get there. Margaret McCarthy, and she might be somebody that we could think of inviting to come and visit us because she's really, she's an impressive speaker, a very entertaining speaker, and she's already stepped over that boundary. And her slides often, towards the end of the talk, get into a lot of interesting topics that I'm clearly not going to go into at all. Everybody in the audience has seen 100 pictures of an oligodendrocyte or a Schwann cell putting myelin down on an axon. And I think everyone in the audience would be prepared to have a short conversation with me about the value of myelin. Why do we want to have myelin? What's its purpose? Just remember that the giant squid axon, which is about 500 micra in diameter, conducts at about 25 meters per second. Now, that's kind of fast, but we can take an axon that is 1,000 times smaller in diameter and put myelin on it and have it conduct at twice that velocity. So the invention of myelin by evolution put us in a position to be able to communicate enormously more quickly and at a tiny fraction of the expense. You know, this was the first move. Whoever invented this would have been a person who really could have been part of the Green Party. This is an unbelievably economical way to send neural information. Now, you want to have it every way. Some axons might be better with a built-in delay, so you wouldn't put myelin on them or make the myelin thin so that it will conduct more slowly. You have tons of options, and that's what myelin gives us. It wasn't long before these pictures appeared over and over and over again, and everyone had their own little jelly roll notion about how the myelinating glial cell put those wraps around and then would squeeze all of the cytoplasm out to get hard, compact myelin. And I would say up until the publication of a paper a year ago, there was absolutely no consensus about the 2 or 3 models that have been proposed. But in an absolutely brilliant piece of work led primarily by the senior author, Michael Simmons, who works at a Max Planck Institute in Göttingen, I think the dilemma is over. Now, before I say anything more about how this all comes about, I want to remind everybody that you have a lot of brain that gets myelin. If you move up the phylogenetic scale from a mouse to man, there's a nonlinear expansion in the amount of your brain which is white matter, which is myelinated tissue. It increases in an extraordinary fashion, and it points out simply that in a mouse, it does make sense to primarily focus on gray matter because 90% of that animal's brain is gray matter. But by the time you get up to the people sitting in the audience, then you have to pay a lot of attention to white matter because it is geographically a gigantic portion of the brain, and it appears to have been endorsed by evolution as one of the ways we can go from running around, collecting little pellets to being able to give lectures and accomplish the wonderful things that have come from civilization. So that's my pitch for white matter in general. The way myelin gets applied and distributed is stunningly complicated. We don't have all the answers, and I'm not going to give them all to you today. Bear in mind that myelin goes on many axons very early in life when we're about a third of the height we are today, and then your limbs grow and your nerves grow and the myelin intervals grow, and all this has to be done in a consummately perfect fashion so that you're coordinated and you're able to master all the motor skills that you need to. This is obviously a cartoon with a couple of pictures, and I'm using it simply to make sure that there's no one in the audience who would be confused about the fact that an oligodendrocyte in the central nervous system gives off 15 to 25 processes. Each of these processes is attached, affiliated with an internodal piece of myelin, for example, here, oligo, and then this internodal piece of myelin. And during the lifetime of that oligodendrocyte, it needs to maintain that myelin sheath. A really clever experiment was done using radioactive signals that came about from the time of nuclear testing in the 40s, 30s, well, actually not 30s, but from the 40s. And tiny amounts of residual radioactivity can be traced, and it's permitted people to discover that most of the oligodendrocytes in your brain came about at about the time you were born. and you still have them today. You're constantly making new ones. I'll make that point again in just a moment for the third piece of this conversation. But the myelin lasts essentially a lifetime. Whenever you see a drawing like this, it grossly distorts what the axon and myelin really looks like. This is courtesy of Bertil Hilla, and it's a good-sized axon, and that red arrow points to the node of Ranvier. And this should impress you about how incredibly minute the node is in comparison to the length of internodal myelin. It's a monstrous disparity. Okay, so this is the paper that I'm referring to, published in 2014 in Cell. It is wizardry, modern wizardry, using a variety of genetic techniques and some of the most powerful imaging techniques that have ever been invented, including this, this pressure, help me here, Gwen, pressure frozen or pressure fast freeze procedure for fixing the tissue, permitting you to look at electron microscopic level tissue at that level of resolution with tremendous tissue preservation. The pictures are nothing short of stunning. Using all these things, he's developed a model, and he's always cautiously calling it a model, but I think the case has been made in a very definitive fashion. So rather than drag you through all of that, I'm going to show you what the model looks like. When you start, the oligodendrocyte gives off its long process, and it makes a paddle-like foot. And that paddle-like foot has a growth area shown right here in pink. And that's the only place that new myelin, that new tissue is being formed. It's a magnet for the components of the myelin sheath, which are not elsewhere in the membrane of the oligodendrocyte, but wind up ultimately incorporated into the myelin itself. This little shovel then becomes applied to a neighboring axon. And this cross-section shows you that, that when you cut through this, you see that at the very start, it cuddles up to this, makes a very close, intimate contact, and begins to curve around the axon. Things continue. The leading edge, this part right here, this spade-like thing, burrows under the tissue that it encounters. And it keeps growing, adding with little motors, more and more structural protein to the membrane, and burrows deeper and deeper, and is driven by this addition of membrane to poke itself under the overlaying membrane. At the same time, there's growth at the edge, and we'll come to that in just a moment. So this goes on and on and on until you have anywhere from 5 to 30 wraps of myelin. Now I want to point out that the design of this is quite brilliant. There are cytoplasmic channels that the oligodendrocyte uses primarily during development to ship nascent protein building blocks up to this little spade-like beginning of the myelin sheath. These channels, which in static histologic sections are called, they're called the clefts of Lantermann-Schmidt, or Schmidt-Lantermann clefts. And these are very present during development and then gradually disappear. So you can see how this goes, and ultimately, while this is being driven forward, the sides are moving out. So it has this spade-like configuration, and then late in the myelinating process, it moves out so that it now looks like a leaflet or a page of an open book. While all of this is happening, the membranes are accumulating on the outside edge and are gradually compacted. They overlap one another and they get pushed further and further down and ultimately take the configuration of what we call the perinodal loops. So here's another a bit of a cartoony version to bring me to my final point. The compaction of myelin, the squeezing out of all the cytosol to make a truly electrically insulating membrane around the axon, takes place from the outside in. The first bit of compaction takes place at the very outside, and then the last place that you're going to get this compaction is right next to the axon. So that's pretty much the story. I could not resist showing off for Michael Simmons this beautiful, beautiful picture. So here you've got, using this new fixation technique, just a brilliantly clear picture in cross-section of a myelinating axon. You can see that it's progressed. Here's the inner tongue process that has been added to, and spiraled around and around and around till it finally has created all of these laps of myelin. And then from outside in, you see that the cytosol is gradually squeezed out, and these last loops towards the inside will be the last ones that will have their cytosol squeezed out. I want to leave you with a final thought, and I'll return to this towards the very end in about 15 minutes or so. Here's what things look like at the end. If you were to unwrap all of that myelin, you'd see something that looked like this little paddle, which is more or less a perfect rectangle. There is cytoplasm all the way around it. The cytoplasm at these edges, of course, will go into the perinodal loops, and the inner tongue process, shown here in an electron micrograph, will be this front loop. So there is always cytosolic continuity between the cell body and between these edges around the myelin. If something happens to your myelin, let's say that you have an injury, and part of the injury involves white matter, and part of it involves a partial demyelination. Your nervous system will make an effort at rehabilitation. It will try to restore that myelin. It begins to more vigorously express genes that had been turned off, like myelin basic protein and others. And then it attempts to ship those down so that some myelin repair can take place. As that begins to happen, there is a process where a lot more of these cytoplasmic channels are created, so that these far distant reaches of the myelin, places that have always puzzled anatomists and physiologists alike about how is the oligodendrocyte able to have any communication whatsoever to something that looks impenetrable. It is never completely impenetrable, and if there is need for more rapid myelin synthesis, then these channels reform, and it gives the cell body and the genetic machinery, the protein manufacturing machinery, access to the myelin. Staying on the theme of yes, please, Tom. Two questions.
One is,
do I hear three questions?
Is it the same for oligos and
No one knows that, Tom. No one knows that. The picture looks surprisingly similar, and my suspicion is that this incredibly anxious young guy, and he's very young, Michael Simmons, who's just become a professor, he left the Max Planck this last summer, at the beginning of the summer. I suspect he must be looking at this. When you look at the work, it's almost, well, let's put it like this. A simple physiologist like myself could never imagine the painstaking labor that would be involved in doing all the freeze fracture and the three-dimensional reconstructions and the genetic modeling, et cetera, that this guy did. But I'm sure he's going to do it in Schwanson.
The second question is, during the compaction process, you can't visualize where all that cytosol goes. So cytosol is squeezed out to where?
Out to where?
Where does it go?
Yeah, well, that's an interesting question, Tom. You know, it is funny. Sometimes it does get down to that simple mechanical thing. You just can't figure out where does it go? You know, where does that go? No one knows the answer to that, Tom. It happens slowly. My suspicion is that it simply redistributes probably out to that cytoplasmic gutter and perhaps you know, to a few of the residual Schmidt-Lanterman clefts so that it gets back to the cell body. But I don't have a precise answer. Yes, Cliff. You know, I think it gets less efficient, and certainly some oligodendrocytes die with aging process. But the oligodendrocytes are doggone tough. In fact, the marvel is that even though we were designed, basically we were propelled during an evolutionary period when none of us got to live much beyond age 30. Here we are, almost everybody beyond age 30, and some beyond 2 times that or even a bit more, and we're doing pretty doggone good. So a very robust design. I think it's German probably originally, but I don't know for sure. Okay, so I want to take you in a slightly different direction. If you have never heard William Richardson, give a lecture, he's a Brit, and if you first visited with him in the hallway, you might decide that that's not something you want to do because he seems a little halting and I don't know, just sort of you know, a British reluctance about things. But in front of an audience, he is profoundly entertaining. And the story related to this science paper, he gives in a fashion that, well, fortunately, you haven't heard him speak, I don't think, so I'm in pretty good shape. But unbelievably entertaining. So I'm just going to give you the highlights of what I think is a very important recent observation by Bill and his group. Bill was interested in the idea, based in part on the fact that if I take John and I invite him to come to my juggling camp, and John tells me that the last time he tried to juggle, he hurt himself badly because he got to 2 balls and the baseball hit him in the head. And so he's reluctant. But I say, John, look, I've got the best jugglers in the world. I think I can get you up to 3 balls. And John, we might even try for 4 balls. So he says, okay, I'll do it. I do an MRI on John before this juggling adventure. And then I come back and repeat that MRI in about 2 months after John's really mastered it. He got up to 4 balls. He wanted to go to 5, but I took a good look at him. I said, John, stay with 4. And when you do that MRI, a number of people have had the interesting, slightly subjective, but objectifiable, as much as you can be with MRI, the only thing that's ever been seen is a change in myelin. It's the only thing. And people have seen it with juggling, they've seen it with piano playing, they've seen it when you learn a new language, sometimes in different places, but always in white matter. So Bill said, there's got to be something going on. So here's what he did. He said, I'd like to take and get some genetic models where I can prevent John from being able to make any myelin. And I want to be able to do it when I want to do it. So I'm going to make the turnoff of a gene called, I always forget, it's called, it is called the myelin, give me 1 second. It is called the myelin regulatory factor. The myelin regulatory factor is a transcription factor. And if it doesn't turn on, then you'll never get an oligodendrocyte. You can have tons of these in our brain. About 3% of our brain is oligo precursor cell. It depends. Sometimes 3, sometimes 5. Depends on the area, depends on age, a few other factors. So 3 to 5% of your cells in your brain right now as we're sitting here are these precursor oligodendrocytes. They can't make myelin. They're expressing no myelin genes. They're sitting there. A small number of them, over your entire lifetime, convert to oligodendrocytes and begin to make myelin. We've never paid any attention to them because the majority of your oligodendrocytes were born and made when you were a baby and then have hung out and made adjustments during a lifetime. But you've got these interesting precursor cells. So he said, I want to make an animal where I can stop them from converting to oligodendrocytes that are myelin competent. And he did just that. He could turn it on with tamoxifen. He'd give the animals, which grew up completely normally, tamoxifen, and now suddenly they have a genetic expression pattern where in these precursor cells only this gene goes off. They will never become oligodendrocytes. They will never express any myelin. And here's how impressive it is. He gives them that drug, and he gives it to two animals. One which is not going to have this effect, the myelin, what do I call it? Myelin regulatory factor. Let's just call it MRF from now on. So in this particular one, the blue line, it's a control animal that the gene will continue to be expressed. The red animal, it's done. And when he looked over a 3-month period of time after starting tamoxifen, you can see that there is this, and it looks like a big steep line, but I can tell you it's in the neighborhood of a few hundred new oligodendrocytes able to make myelin, and nothing at all in the control animal. So you could see this plain as day. Green is myelin here, and it's the same experiment essentially, except we're not counting cells now. We're looking at myelin. This is all brand new myelin over a 3-month period of time. And this is the animal that's got this gene repression, no myelin. So no oligodendrocytes, no new oligodendrocytes, no new myelin. Old oligodendrocytes are absolutely perfect. Nothing's changed. And if the animals, if Tom behaved exactly as the animals, I could turn that gene off in Tom, and for the next 10 years, he'd never know I did anything. Well, unless he wanted to juggle. We'll come back to that. No. Yes?
Those are heterozygotes. Is there a difference between homozygote and wild type? Or they're haploinsufficient for that transcript?
Yeah, there's absolutely no difference between the wild type and those heterozygotes. So myelin's completely normal. Their behavior is completely normal. Everything that they had learned, they did some experiments where they taught them things. Everything was totally preserved. They were terrific. Let's see, one other little thing I wanted to point out. It's the same thing. This is the time to fall from an accelerating rotor rod. It was unchanged compared to control. So the motor performance was completely normal once you turned off this gene. Now this is interesting. It turns out that Bill Richardson's an expert on mice athletics. And he knew that mice love the running wheel. And what you don't know is how far a mouse will run if given access to a running wheel over a single night. A little tiny mouse will run between 5 and 7 kilometers. It's so stunning. The first time I heard it, I was certain there was something wrong with the acoustics. But he's measured this in every which way you can imagine. So these animals are crazy. They run just like nuts all night long on these things, and they love it. That's their preferred thing to do. Now notice this. This is a nice little running wheel. I think I could do this. It's got little pegs in every single spot. He messed them up terribly. In the dark, he's given them a running wheel that has pegs missing. And the mice don't like this at all. They get on this, they fall off, they complain bitterly, they talk to the neighbors, what happened, what's going on here, poor manufacturing of running wheels. But they like to run, and if you give them enough time, they get doggone good at it. They have to adopt a brand new motor strategy in order to do it, but they get terrific. So that was the chore. to acquire this new, more difficult task, and they do it well. So the experiment was to put them in this complex wheel and give the heterozygote, the homozygote, tamoxifen, wait 3 weeks, and see how they do, see whether they can acquire this skill. And so that's what they did. And what they found is that no matter what they measured, average speed, the number of times they fall off, on and on and on, inevitably, the animals that had this incapacity to make new oligodendrocytes or make myelin never got very good at this task. They never learned it. And you could do it as long as you wanted to. Then he decided, well, what's going on? What's happening with the oligodendrocytes? So he made some measurements of first of the precursor cells, which are identified by being positive for platelet-derived growth factor receptor, which identifies these precursor cells. And he found, here's a non-runner. So here's some mice that are just sitting around. They're sedentary mice. They like the wheel, but they don't get it. And there's inevitably, after a couple of days of running, a very substantial bump up in the division of these precursor cells. So after 2 to 3 days of running, boom, there's a big bump of the precursor cells. I'm not showing you this, but 4 days later, there's a big bump in mature oligodendrocytes that begin to express myelin. So running on this complex bar induces the creation of new oligodendrocytes and new myelin. And I guess actually I do show that here. So these are new oligodendrocytes running in red and not running over here. And of course, this is out a little later because the first bump is in the precursor cells. So where are we with this? And it takes a little while for this to sink in. These animals, oh, by the way, you could, just as a control, to make sure that this isn't a motor performance thing, he took the same groups of animals and he taught them all before the tamoxifen how to run on the more complicated running wheel. And they all acquired the task. Then he gave the homozygotes the tamoxifen and put them back on 3 or 4 weeks later with the controls. Everybody was fine. So they have it isn't a motor performance thing. Once they'd acquired it, presumably making new oligos at the time and new myelin, they had it forever. They could get back on it and go like crazy. So I think it is probably fair to say that what this is telling us is that certain kinds of motor learning require the generation of new oligodendrocytes and new myelin. I think this is a stunning observation. It's something that has sort of been completely submerged as a concept up until Bill's paper. There was just some little soft junk in the literature that I think people thought about as noise and not anything real. Bill's pursuing this. I've seen some preliminary stuff which follows on with even stronger data. I think this is the real deal. I think it probably means that neural activity induces, through a pathway that isn't entirely clear, certain signals to neighboring precursor cells to get them to divide and then to get them to strategically myelinate axon pathways that you need for this behavior. Okay, so yes, Rusty. Yeah, so a good question. Rusty asks whether or not this effect is specific in different pathways. They primarily did measurements in subcortical white matter and corpus callosum. I'm a little fuzzy, Rusty, but I believe that they looked to see whether there were any changes in myelin or anything else in the hippocampus and didn't see it. And the presumption is that this is probably cerebellar pathways, basal ganglia pathways predominantly, and the things that hook them up, since it's obviously something that's going to require corpus callosum. It's a, it's a bilateral kind of motor task. And Tom. Yeah, I mean, ultimately, the excitement about everything we talk about in preclinical is that it's relevant to the audience, I'm, I'm very convinced by this data. It would be nice to move it up the phylogenetic scale. I don't know quite, Tom, and this would be more in your bailiwick, whether or not it might be possible to design some kind of a human experiment to sort of get at this. If we had a tracer that marked newly minted oligodendrocytes or showed up early wraps of compact myelin, I mean, that we could nail it probably. And those things are probably coming. Right? They're just not on the shelf today. Okay, so very briefly, how do central nervous system axons eat? And we've talked about axons. George Bishop was a hero of mine. He worked with Erlanger and Gasser, who won a Nobel Prize in 1926. George was a gadfly. He could be annoying as all get out. He used to torment the graduate students at Washington University in St. Louis, but he was also incredibly lovable. And he stuck by the axon. People told him, it's so boring, et cetera, but he made an incredible career out of it. And as a parting shot, when he was asked to commit something to the proceedings of the Royal Society or some such place, he offered this doglet. And yeah, that's just fine with me. So here's a picture I probably showed you with I've showed you many times. A blood vessel, astrocyte, or rather endothelium, astrocyte end foot, which plasters this completely. So glucose could certainly get into the astrocyte very conveniently. This is not a tight junction across the, across this layer of end feet. It can, glucose can easily diffuse out, get over to an axon or a neuron. We know that energy metabolism includes glycogen. It turns over very rapidly. It's exclusively contained in astrocytes. The astrocyte dumps tons of lactate into the extracellular space, and that lactate can be used as an energy metabolite. So we know all of this. What we don't know is when it operates. We don't know, especially when it operates in all of you. Boy, I'm telling you, this has been a topic of enormous interest for quite some time. Now, just looking at the axon, I want to remind you that the axon has to fend for itself when it comes to energy. The axon gets no energy, no help whatsoever from the cell body. So Claire's axons, the ones that are myelinated, have got to take and get glucose and oxygen from the neighborhood, wherever they are. If they're in her arm or leg or in her spinal cord, that's how they operate. They're not waiting for ATP to come down from the cell body. That's just completely nonsense. There was a time when people talked about that, but it's just, we know that's simply not so. So there's the axon. It's pretty doggone hungry, even though it's efficient, and it needs to make ATP from some substrate. And how does it go about doing this? I only have one other slide besides this picture I've got right here. And so let me just say to you that over the last 3 years, pictures like this one, which are derivative of pictures like this one from Klaus Nave, have begun to suggest that rather than glucose or lactate being taken up by the naked axonal membrane at nodes of Ranvier, where we know there are glucose transporters, by the way. We know there are glucose transporters. We know that there are lactate transporters there. Rather than this being the only place where glucose or lactate might come in, it has become unbelievably popular to think that somehow or other, glucose and or lactate get in the oligodendrocyte and then get distributed by those beautiful cytoplasmic channels we talked about and make their way to the intermes axon and feed that huge long length of internode. So rather than, remember that picture I showed you of Bertil Hilla's dissected frog axon, where an internode was, I forget what the fraction is, it's 0.0001% of the surface area of the axon. So rather than expecting that you're going to get every lick of external nutrition you need by taking it up at that tiny node of Ravier, which looks perfectly possible in this cartoon, which is profoundly out of scale. People have begun to talk about, well, sure, it's got to be, the oligodendrocyte's got to be pumping it in here. Now you might say, well, why can't it diffuse down here? It starts here and then it diffuses in along the axon. And of course, you know the answer to that. Myelin doesn't work unless those wraps are tight. It has a death grip on the axon at the edge of the node so that nothing can get in. Nothing gets in, nothing gets out. So the only deal is it either comes in here across the naked membrane or it's got to be delivered to the intermes axon, which would mean a trip all the way through the oligodendrocyte and then into that little space right around the axon. So why are we talking about this? Here's why. About 3 years ago, Jeff Rothstein, a neurologist, just like many in the room, who's extremely interested in ALS, started looking at some ALS tissue and discovered that monocarboxylate transporter, the lactate transporter, was gone in the spinal cord right next to motor neurons. Okay, so what? So he looked at it and he didn't think too much about it. Then he started looking more, and a bunch of people in his lab started doing cellular stuff, and they realized that monocarboxylate transporter is gone only from oligodendrocytes right in the vicinity of motor neurons. And it appears as if it's just in those bundles, those motor axons, as they're leaving the cell body of the motor neuron. So Jeff said, well, okay, that's kind of interesting. It's just an association, probably doesn't mean anything. It's possible that they get sick because the motor neuron's dying. He did a study looking for the presence of monocarboxylate, MCT, monocarboxylate transporters, and he found that the only place he could find them was in oligodendrocytes, in spite of the fact that myself, dozens of other labs have seen them expressed in astrocytes. So once Jeff published that paper in Nature, and Klaus Nave, another very high-flying scientist, regularly publishing little reviews in Nature and Science, like where he published this. This is in Nature. Started drawing diagrams like this. And I bet that I have seen 20 of them, each one a little more certain than the next. So the evidence is completely circumstantial, and I'm putting this up so that you can defend yourself. The glucose comes in across the capillary endothelium. It's in the extracellular space. It's taken up by all of these cells, axons, oligos, astrocytes. They all have transporters for glucose. Astrocytes are the only cell with glycogen, and it remains to be seen whether or not the oligodendrocyte actually transmits either glucose or lactate, and really what we're mainly talking about here is lactate, to this intercellular space in the internode. But if I didn't prep you this way, when you see this diagram, it's going to be so attractive, and the likes of Jeff and Klaus whispering in your ear about the correctness of this is going to make you want to buy it. Just buy it, the whole story. I don't want you to do that. Okay, I'm done, and I sure appreciate your forbearance for what was a bit of a herky-jerky conversation with you today, but you know, if you weren't here for my opening disclaimer, my computer blew up about 4 or 5 days ago, and although it was sort of backed up, it wasn't completely backed up, and it left me a tiny bit in the lurch. So, thank you for your attention.